for the nice presentation and thank you for the organizer for the interesting program, for the opportunity for me to be here to present this work today. As the chairman, Mr. Chairman said, I come from Lisbon, from the Institute of Experimental Biology and Technology that is very close to Lisbon. And this is a non-for-profit institute that aims the transfer of technology to the industry. And we have as partners the major companies in Portugal related to agro-food and pharma industry. And also we have as partners very well-known pharmaceutical companies as Merck, Bayer, Novartis, that have at the moment satellite labs in our institute. Our aim is to put value on our clients and partners, and doing that through the development of innovative products and novel technologies that can be implemented in their sites. Within this, the laboratory that I coordinate at the moment aims the development of green and clean technologies for the formulation, the extraction of bioactive compounds, and also the formulation and of adequate delivery systems. Today, I choose to bring here the work and show you some results that we obtain within the work that we have been developing concerning the production of delivery systems for the drug delivery to the brain through the nasal administration route. As we all know in this room, the blood-brain barrier is one of the most difficult to cross due to the tight intercellular junction and also to the different efflux pumps. And so the majority of the drugs face several problems in being effective, and this is one of the main reasons, I believe, for the failure in treating neurological diseases. As an alternative, the nasal route can be really a solution, as it can be considered a non-invasive way to circumvent the BBB and faced as an effective approach to drug delivery to the brain due to the peritoneal and trigeminal nerves connection between nose and brain. To develop these systems, we have a challenge because for this we do not want very small particles, so the particles, otherwise it will be inhalable and reach the lungs instead of the brain. And so the particle size should be larger than 5 micrometers. It is good that we have prolonged residence time with high local drug concentrations. Drug deposition in the nasal cavity is another aspect to take into account. And mucoadhesive and haloporous systems are preferred. With this in mind, we have been developing hybrid delivery systems by combining polymers, natural and synthetic polymers with lipids. And so our aim is to produce particles that can take advantage of different properties of different materials. As I mentioned before, we are a technological group, and for materials processing, we use not only what we call at home the conventional techniques like spray drying, spray chilling, or fluidized bed spray processes, but also we explore very deeply the high pressure technology where we are really experts. And using this technology, we have implemented several technologies for particle formation, exploring the different ways of using supercritical fluids. And we can do impregnation by adsorption using also supercritical fluids. And very recently, we implemented also a process to develop aerogels Vanessa that that gave a talk yesterday talk about that. And also we can do molecular imprinting through this technology. In the lab, we go from the fundamental studies to cytotoxicity evaluation. We have implemented cellular assays to do that, and we can do a deep characterization and after the design and production of the delivery systems. I will focus on this technology and how we explored this for this project. For the ones that are not familiar with supercritical fluid precipitation process, I want to start by saying that a supercritical fluid is a fluid that is above its critical point, and normally we use compressed gases to do our process, and the elected one is carbon dioxide. Why? Because it's non-flammable, it's non-cost, it is not expensive, and we are recycling, and it's not toxic at all. In this region, in the supercritical fluid region, the compressed fluids have very interesting characteristics that are always looked for a solvent, because they have density and solventing power similar to liquids, but the diffusivity and viscosity is similar to gases, and so this is very good for a solvent. All the processes are considered clean technology, as at the end we obtain solvent-free products. As the critical point for this for the elected solvent, compressed gas, is quite low, because it's the critical point is 31 degrees Celsius and 74 bar, so quite easy to attain even in the industry. So we have the possibility of processing thermolabile molecules, and at the end, and related with the precipitation process, we normally obtain, or it's easy to obtain, porous structures that take advantage of that. Related with these projects, and specifically for nose to brain, I will show you three different case studies. One that combines a polymer and a lipid. Another one that is a mixer, is a it's a structured lipid particle, and the third one that is alginate-based aerogels. In the first two cases, we are using a technology that where the supercritical fluid is used as a solvent media, and we will take advantage of the interaction between the solvent and the carriers. And in this one, the supercritical fluid is explored as a drying solvent, and also as a media for the impregnation of the particles that are formed. Related to the first two examples that I will show you, the process is called the particles from gas saturated solution. Gas, I will prefer to call this supercritical fluid, because we use supercritical fluid conditions, but this is the name that is registered, so we use this. And the principle of the method is quite simple to understand, because the compounds are melted in a high pressure vessel, taking advantage of the fact that mixing it with supercritical CO2, this molecule rapidly diffuses into the carriers or into the materials, and we observe very significant materials properties change, like plasticization and viscosity, and so we can at the end have a lower effect on the TG and TM of melting temperature of the materials. How do we produce the particles? By the rapid expansion through a nozzle here, normally to the ambient pressure, and also we can have here a drying current by air, compressed air, or nitrogen, or pure nitrogen, or whenever the process, the needs of the characteristics of the final product. The first system that I told you that I will show you the results is the combination of a polymer, in this case PEG 4000, with Gelucir 43. They are immiscible carriers, materials, because this one is hydrophilic and this one has a very low HLB value, so it's a lipophilic carrier. So there was a challenge to produce this. Why did we choose these materials? Because for the purpose, for the final application, we wanted to have biocompatible and biodegradable materials, and this one is an hydrophilic polymer, as I told you, has the property of being mucus penetrating, is non-toxic, and this one can promote the penetration and absorption into the epithelial cells as a lipid, and it is a mixture of, this is the commercial name, but this is a mixture of mono-D and triglycerides with this chain length. As I told you, we always do the fundamental studies to better design the processes and to get faster to the results. And in this case, the challenge was the mixing of the two materials, and we overcome this by producing the emulsions. So what we put in the high pressure vessel, or what we produced in the high pressure vessel, is the emulsion, but the emulsion of the two materials additional with the addition of the supercritical CO2 that mixed in the mixture. So we had to, with a visual cell, a high pressure visual cell, to study the equilibrium time, because the control parameters that we have for the process are essentially the equilibrium time, the pressure, the temperature, and also the ratio of the carriers and the solvent. So we studied the exact time that we should do the mixture to have and simulate the expansion to have a stable emulsion. Otherwise, it will be a phase separation, and the product will not have the characteristics that we wanted at the beginning, because we wanted to have core shell particles with Gelucir inside and PEG as an outlier. After producing this, we characterized, and using SAM and FIP, combined with FIP, we were able to see the particles and to cut the particles, and what we saw was that this is really the mixture of the two materials, as we have this kind of water falls inside, characteristic for the most soft materials, in this case Gelucir, and the outlier seemed to be PEG. To confirm, we do TEM experiments using a dye for the lipids, and in fact what we got was not a core shell as what we were intended to have at the end, but a matrix. We think that we have a matrix of PEG with several cores of Gelucir. The combination of the materials in the same particle was confirmed by the release profiles using a model drug to follow the release, and in fact we obtained release profiles that represent a merge between the two carriers. This work is still ongoing, because now we are optimizing the conditions to have smaller particles and also avoid disaggregation and try to have single core shell particles. The second system that we studied was the combination structure lipids, so the combination of a liquid and a solid lipid in the same particle, and through this way to have higher loads, and also to the GMO, the liquid lipid that we choose, add mucoadhesive properties to the system, additional mucoadhesive properties, and the Gelucir, as I told you before, was there to increase the uptake by the epithelial cells, and the combination of the materials allowed the increasing the stability of the final product, because if we have only solid lipid particles, they are not so stable. The fundamental studies consisted in evaluating which is the temperature that should be used, because these two materials really are affected by the presence and the solubilization on CO2 into them, and we observe a very significant decrease in the melting temperature, so this means that we can handle and process even more sensitive drugs, because the decrease in the melting temperatures is quite significant, and so there is a very huge difference between using this process or using, for instance, spray drying, but with the melting device to spray melted lipids. Spray chilling method. Well, the principal results were that we followed the stability during storage, and we saw that the hybrid system was much more stable, and it allows higher ketoprofen encapsulation efficiency. Again, this was a model drug that we used. It's important for us to follow the kinetics of release, and so we have to use the drugs. Also it has higher permeation through mucous membrane model, and we reach really good efficiency encapsulation in both cases, but higher in the case of the hybrid system. Those two examples are already published, and we have the reference in the presentation. The third case study is the production of an aerogel, and as I told you here, the supercritical fluid is used as the drying solvent, because it was already explained yesterday, but to produce this aerogel, we pass it through the first step is the production of the hydrogel, then we have to replace the alcohol by the water by alcohol, and then to dry the alcohol, in this case in a clean way, and it was using CO2 that dissolves the ethanol that was used. The final products that we usually obtain in this process, in these aerogel productions, are amorphous solids of low density, porous structures with large surface area, which allow the high drug loading most of the times observed. Up to the time, the aerogels produced were in the form of monoliths, beads, or microparticles, but with sizes not below 50 micrometers. In this work, we were able to reduce the size of the particles by modifying some aspects in the process. It consisted in mixing an aqueous solution, in this case containing the polysaccharides and calcium carbonate, and emulsify it with paraffin oil, and then by adding the an acidic microemulsion, we were able to obtain at the end very low size aerogel microparticles. We loaded this with ketoprofen and quercetin as two examples of lipophilic or amphiphilic and hydrosoluble drug. The ketoprofen was loaded at the end after the particles are formed and using supercritical CO2 impregnation. So in this case, the supercritical technology was using to put the drug inside also. And the Kirstein was loaded during the aqueous, during the ethanol, the alcohol gel production. At the moment we are trying also, it's a challenge to put, to develop aerogels containing small peptides. I'm almost finishing. The particles that we obtain, the alginate-based hybrid were in this range of sizes. So the first alginate pectin system that was explored has the highest surface area and the lowest particles, or the smallest particle size. Both of them very porous and spherical. And at the moment we are doing cell-based assays, and they showed to be non-toxic. I will pass this. As conclusions, main conclusions, with the PEG gel-usir systems we obtain multicore lipid shell polymer particles produced by PGSCS, but with large size yet, and with a high polydispersity that we want to improve and be less dispersive. The second system, we obtain structured lipid particles, also using this technique. They showed to be more stable. We were able to load it with significant quantities of drug, of the model drug, and we observed an enhanced permeation compared to solid lipid particles. The last one was the most promising, and we obtained really small, but yet in the range of size that we should have the particles for the purpose of this work. And these particles are highly porous, and we expect to have fast drug release. So it is the most convenient systems. I want to acknowledge my group, mainly Vanessa Gonçalves, that is the PhD student that developed all the work, and also the funding institutions that support this. Thank you for your attention.
And thank you for your presentation. We have time for one quick question, if there's anyone.
May I take the pen?
About the nasal route of delivery to bypass the blood-brain barrier, what is the limitation? I mean, your particles only serve as a release depot for the drug, which then gets into the blood vessels in the brain, right? And is there any limitation for, I mean, how leaky or how easy it is to get any kind of drugs in the blood vessels in the brain, bypassing the blood-brain barrier, using the nasal route of application?
Yes, I think the strategy is to have the higher load as we can. And then, we know that the quantity that reach the brain is not really a huge quantity. So there is a huge difference between the load and the quantity that really reach the brain. It's why we think that these systems maybe are most promising for the very effective drugs that need not so huge quantities to reach the brain, no huge concentration like active peptides. So we are putting our efforts in trying to load them with these kind of drugs that are much more effective.
Well, thank you. We are a little bit short on time, so let's leave other questions, but not before you get your certificate, which you are here.